Hello, everyone. Good morning. Thanks, Kristen, Sandy, for allowing me to speak today. I'm kind of following on from Andrea's talk yesterday a little bit, but zooming way out. So she spoke about, as a reminder, T-cell radiomics using some of this data that I'm about to show you. But I kind of wanted to show you guys like how it is as a mathematician being in the operating room and kind of like the steps that we take to collect this data, and then a couple of things that we can do with it. So this is a brain tumor biopsy protocol that we kind of coin inside internally. The biggest thing that really I wanted to think about, like, there we go. The biggest question we're trying to answer really is how can we connect tissue biology to imaging on sort of a local coordinate level? And we believe that we can do this through the collection of image-localized biopsies. So I'm going to kind of break this up into like what we have to do before surgery to consent the patient, all that stuff, during and after, and then a little bit about what we can do with the data. So before surgery, first thing we have to do, right, is identify patients. Like that's obviously we need the patients. That is actually quite a lot of work because we have to check the electronic health record, see who's on the surgery schedule, see if they're appropriate patients for us. Say we have a patient, we have to then communicate that with the clinical research coordinators. They have to then reach out to the patient themselves, explain the study, explain everything that goes with it, and hopefully the patient agrees to consent to the study. We then have to reach out to the people who are taking the MRIs because we also try and protocol for some advanced imaging, which I'll explain in a second. Then we have to say, okay, well, do we have the people to cover this surgery? Like, for example, right now there's a surgery happening, but I'm here, so like I couldn't go. Communicate that with clinical staff and then show up for the day, on the day. So a bit about the advanced imaging. So with brain tumors, typical images, there's T1 weighted with contrast. There's T1, T2, and flare. That is kind of typical MRI sequences that we get. If we catch a patient early enough and we can protocol for our advanced imaging, we also aim to get regional cerebral blood volume, dynamic susceptibility contrast imaging, and some diffusion weighted or diffusion tensor imaging as well. Like Andrea said yesterday, like the imaging doesn't show really the complete picture. So we hope that with more pictures, we can get a little bit more insight into that story. So here's kind of a workflow from left to right of like what happens during surgery and then kind of after surgery. I walk into the OR, I'm in scrubs at that point, and we have this machine, it's called a Stealth on the left there. It's created by Medtronic. They make a bunch of medical devices, and that is for intraoperative neuronavigation. So the patient is registered to their most recent MRI before the procedure starts. So the surgeon can use a tool to say like, okay, well, I'm here in the brain, where am I on the MRI? So then that's already the clinical workflow. So we come along and we say, hey, when you collect the piece of tissue for us, can you use that tool first? Say, this is where I am and this is where the tissue came from. And we can track that on the Stealth machine as well. So we end up with XYZ coordinates for all of these samples. We aim for 8 samples per patient. We aim for a reasonable spread around the tumor. So we can kind of capture some heterogeneity that's typically present. And then we flash freeze those samples in the operating room in liquid nitrogen. If a sample's big enough, we've recently started also adding them to formalin for FFPE as well. And those flash frozen samples, they go through whole exome, bulk RNA-seq, and that data all gets thrown down the pipeline. But also we have to track inside the operating room all the samples that came out, even the clinical samples that came out, because it's really important that all the tissue is accounted for, both in the patient chart, with us, and it's all the same, we all agree. All that data then goes into our in-house database, Patient View, which maybe you've heard of before. Kristen has kind of pioneered this and grown it throughout her career. So we have a large database of patients, mostly glioma patients in there. So here's an example patient. This is a 58-year-old male who had grade 4 astrocytoma. They came to us, they got some imaging. We segmented that imaging. Here's a kind of a 3D representation of that. The T1 GAD, which is considered like the core of the tumor, is shown in red here. The T2 in blue. So they come in for their first surgery. We collect biopsies. Again, we can track the XYZ coordinates of those. and kind of spin it around for you guys to see. Then the patient, unfortunately, progressed 5 months later. They came in for a second surgery, so we can update our imaging. We can then also, we're fortunate enough to collect another set of biopsies for this patient. So we end up with a total of 16 biopsies for this patient. Yeah, so just as an example. On the left here, I just want to talk about, although we can look at like higher spatial resolution, we can also take a little step back and classify in terms of, did this sample come from a contrast enhancing region or a non-enhancing region, to kind of group and analyze those separately, because we do expect differences in those regions. Again, these tumors tend to be heterogeneous, and it's important to consider the non-enhancing region because that's typically what's left behind after surgery. The surgeons aren't always able to get everything, especially with like high-grade glioma. So the non-enhancing region is typically what leads or populates the recurrence. So it's important to kind of understand that for the future of that patient's disease. So just a bit about our current kind of counts that we have as of March 31. We have had a few patients since then, like today and yesterday. But so we have approached a total of 246 patients. There is a little bit of dropout. Some patients actively decline. They might just not want us to do research on their tumor, which is completely fair. It's a scary thing to have, and they just want the clinical staff to focus on their clinical job and not touch anything else. Some patients also passively decline. They just might not kind of sign things in time. But we have a total, as of March 31, a total of 203 consented patients. Another source of some dropout is we might get to the operating room and the surgeon says, actually, this case kind of isn't going to work for research because maybe it's in a tricky location. Maybe they're only getting a biopsy and they need all the tissue they can get for clinical reasons. So sometimes that happens too. But we have attended 134 surgeries for glioma, 38 MET cases, and 10 with some other diagnoses. So the tissue that's been analyzed so far, we have whole exome sequencing for 54 patients with 235 samples, bulk RNA-seq for 45 patients with 151 samples. Again, this is an active process. We tend to send these out in batches, so these numbers will grow. Demographics. So on the left here is the histogram by patient age, stacked by patient sex. We do have a reasonably balanced sex split. Thinking back to like Melissa's talk the other day, this is patient-reported sex. This isn't like chromosomal sex. And then we also have patient-reported race and ethnicity. We do predominantly have white patients who identify as not Hispanic or Latino. So it's not the best data set for doing health disparity research, but I'm hoping those numbers improve. We also track patient survival, so that involves going back into the chart, determining if the patient did pass away, or maybe we have lost them to follow up. I'm just showing an example here, split by grade. We can split by whatever we want. And so what can we do with this data in terms of algorithms and more mathematical things? So a couple of things that we're doing, Andrea mentioned yesterday CyberSort, which is a tool that we didn't develop. It's developed at Stanford, but it deconvolves bulk RNA signals using a single cell signature matrix. So Andrea spoke about this data set yesterday that's called LM22. It's got a bunch of different immune signatures. But we've worked with a group at Columbia, Peter Connell's group, who have their own single cell data set, and have kindly agreed to let us use it, that splits, categorizes single cells into different phenotypes of glioma, different astrocytes, and some immune populations. So we can take each of our bulk RNA signals and kind of split that up into here percentages or estimated percentages of, okay, this piece of tissue was 50% like mesenchymal glioma, for example. On the right there, I'm also showing a plot from Monocle, which is a reverse graph embedding algorithm. Basically what it does is it takes the RNA data and puts it on a really low dimensional space and orders in each individual tissue sample by what they call pseudo time, which effectively is a metric for how the progression of the cells. You see kind of in this example, it splits into like 3 clusters. On the left, it's kind of like a normal-ish phenotype. Top right is more an immune dominant, and then bottom right is more malignant, so like that's the glioma. But the novelty of our data, right, is that we have this imaging stuff. So why not also split it by that? So if I split the samples by contrast enhancing and non-enhancing, these are the same plots I showed before, just now stratified by that. We can get further information to see, okay, well, there are differences, for example, in glioma subtypes between these two regions. And like I said earlier, right, it's important to understand like the non-enhancing region, because that's likely what leads to recurrence. And also on the right monocle, we can see that the non-enhancing in red there typically clusters towards a more normal like samples, which makes sense because they're the ones that are further out there, less developed, also typically within a more normal, whatever normal is, environment. There are some limitations to this work, which some of them we just can't really control. So we ask the surgeons every time for a pencil eraser size sample. However, limitations of tumor size, tumor location, just sometimes even the tool the surgeon is using can change things. So I put kind of some extreme examples there on the right. That's my thumb. And a little tiny sample versus a larger sample next to a paperclip, which hopefully gives some kind of sense of scale. Also, there's brain shift during surgery, which currently the intraoperative navigation can't account for. That depends on the patient's kind of position when they're having their surgery, and also the location of the tumor. There's some registration error with the stealth machine, sometimes human error. Like communication is really important in the operating room. And, you know, like I said before, we have to liaise with clinical staff, both the surgeons, circulating RNs, the scrub tech. And if we're getting like 8 samples in a few minutes, sometimes things fall through the cracks occasionally. Also funds for tissue analysis. Like it would be great, right, if we had like single cell data for all of our samples. We have so many samples, that gets really expensive really quickly, especially when we started this in 2017, when single cell was just like way more expensive. Next steps that we're trying to do. So at this point, the clinical staff, the stealth technicians are very well trained at doing this because they're so used to it. So we've proposed, Jasmine, my colleague, has kind of led this, that they could do this in the clinic. Like, why not, right? You already have the stealth there. You're already collecting clinical samples. There's no additional cost to doing this. It's just more data. Us as mathematicians love that. And more data, it's better every time. There's not really much more workload to that. It could be useful for tumor board decisions if they know where locations were, if they have like a recurrent case. And also for pathologists who are looking at these cells under the microscope and they know, oh, this is tumor core. We expect more sparse tumor cells here because it's like way far out. So we've put some of this work into a preprint. Last October, I think it came out in medRxiv. There's a QR code there. It's also currently in review at +1 protocols. If you guys want to learn more information about that, or obviously you can come ask me. And you know, there's a ton of clinical people involved in this work. So there's probably people missing from this list even, but you know, there's pathologists, pathologist techs, circulating RNs, scrub techs, obviously the surgeons, the clinical research coordinators, the stealth techs. It's just a big thing that obviously I didn't do myself. And to coin a phrase that Christian used in our preprint, it takes a village to do this. So I really appreciate everyone who's been involved. Thank you to the funders, obviously the NCI, the Ivy Foundation and James S. McDonald Foundation. There's some photos there of us in scrubs to prove that I do this. And obviously thank you to the patients and their families for like agreeing to share this data with us. And thank you all for listening.
Yeah, I do think this is super. And as I asked yesterday and was referred to today, I guess, have you considered using or looking at other aspects such as, I don't know, mechanical properties of these cells? So beyond the genetic data that you're gathering, which is obviously super important, but I could imagine that you see differences in other parameter spaces as well.
So mechanical, so is in like mathematical modeling applied to these?
No. So that actually taking biopsy samples, right, and doing single cell mechanical characterizations on those, which you can do these days.
Oh, like flow cytometry and stuff like that?
Imaging-based flow cytometry.
Imaging-based flow cytometry, oh.
So where you shoot them through microfluidic channels and then see how much they deform, and you can do like.
We haven't really considered that. The one thing that I mean, I'm a mathematician, so my understanding of flow cytometry is very limited. But my understanding is you're typically targeting a single like cell type when you do that versus characterizing everything.
You can now characterize everything, can do whole blood samples and have million data points in 10 minutes. If you think it's interesting, then come and talk to me.
Yeah, no, I didn't know it had that capacity. I thought it was like a single cell type, and then I was going to say that we're quite interested in generalization, but if that's not a limit anymore, then
real-time deformability cytometry.
Awesome. Yeah, cool. Please come find me. I'll come find you. Thank you.
Nice talk and really exciting data set and tissue set.
Thanks.
How many patients do you have multiple time points and have multiple biopsies per time point?
Good question. So we definitely have like 3 or 4 with analyzed tissue. We have some more, like we just got our first patient with 3 surgeries who was I want to say a metastatic melanoma case, but we haven't analyzed the metastatic tissue yet. So it is limited. Like some patients come in, they have 1 surgery, they might pass away before they reach their second, or we have patients who got a surgery elsewhere and come in, we have one, we start at recurrence kind of thing.
And your average number of biopsies on a time point?
Sorry?
What's your average number of biopsies on a time point?
Our median, it depends on grade. So our median for like grade 4, grade 3 is 8. That's what we tell the clinical staff. It's nice for them to have a number in mind versus coming and saying like, we want as many as we can get, which is true, right? But like,
and you're getting whole exome sequencing and RNA sequencing on each of those 8?
That's the aim. Yeah.
Have you guys looked into any of the phylogenetic methods for estimating evolutionary parameters from that?
We've done, I didn't do it. Our MD PhD student has since moved on, but she did create some phylogenetic trees where like there was something about the length of the branches corresponded to how evolved they were from each other. We did try and look at that and compare that to physical distance that we have from the images. We didn't see strong correlations. I don't know. In my naive mind, I would have expected to.
And we might be able to help with that.
Sorry?
We might be able to help with that, measuring mutation rates and things like that.
Awesome. That'd be great. Yeah, thanks.
Do you look at the primary tumor resection? Like, do you get any of the surgical resection samples from that as well and do the same sort of analyses?
Yeah, I mean, this is primary, weird phrase, but yeah, we mostly do primary cases. Some, like I said, some patients don't come to Mayo Clinic before they're already at recurrence, so then we'll jump in then. But we do track that in data set. I didn't show primary versus recurrent here, but that's something we stratify by a lot.
Sorry, I meant to say because what you're showing were like small needle biopsies, right? But you also take out a lot of tissue at the surgery. Do you get to look at that as well at the same sort of?
So typically, it depends on the case. So a primary GBM, for example, or suspected GBM, they would take some clinical samples, confirm with pathology. But they'll also take what they call clinical permanent stuff. And that is kind of like bits of tissue that get added together and it gets sent off at the end, but it's not tracked where it came from. Yeah. And that stuff just goes to a bank that, like, if they need it for clinical purposes, they have extra tissue. We haven't really utilized that as much as maybe we could. I think it's kind of striking a balance between like not burdening clinical staff and still getting what we need, you know. But that stuff like that does exist. Yeah, but it's not image localized and it hasn't had any kind of tissue analysis that we are using like these ones.
Thanks.
Yeah.